Hi there, my name is Jason Crowell, and I'm a neurologist at Norton Neuroscience Institute. Thanks so much for joining us today. I'm going to be talking about the future of treatments for Parkinson's disease. I don't have any relevant disclosures for today. So I wanted to first start with a brief reminder of some of the pathology associated with Parkinson's as kind of just a background for what new treatments might try to do in the future. We know that Parkinson's disease is associated with low levels of a neurotransmitter called dopamine. And this is a cartoon showing on the left normal amounts of dopamine being released from one brain cell to another. And on the right is a neuron in the brain of an individual with Parkinson's disease. And this is meant to show low levels of dopamine being released from one brain cell to the other. And dopamine is important for a lot of reasons. It does a lot of things in the brain, but one of the things it's primarily responsible for is the smoothness and fluidity of our movements. And so if we have low levels of dopamine, that can be associated with symptoms like slowness and stiffness and even involuntary movements like tremor. So the question for a long time has been, how do we increase dopamine in the brain? How do we make more of this available to help with those symptoms? And this has really been the main focus of Parkinson's treatments for many years. This is another cartoon that shows how different categories of drugs work to accomplish that in Parkinson's disease. There's a lot on this slide, and we're not going to get bogged down in the details, but I did want to just point out a few different classes of drugs that are currently available as background for what new drugs might try to do. So just for orientation purposes, briefly, this red cylinder is meant to be a blood vessel. This is part of the brain. This is the blood-brain barrier. These are two neurons, two brain cells in the brain of someone with Parkinson's disease. And so what the drugs that we have currently available, how do those work? So let's start with probably the best known medication, L-Dopa or Levodopa, also known as Sinemet when it's packaged with carbidopa. When someone takes Levodopa by mouth, that medicine is absorbed in the small intestine. It's eventually travels through the blood up to the brain. It crosses the blood-brain barrier. It's taken up by brain cells and released from one cell to another. And so we know a lot about levodopa. It's been around for 50 years, and it's extremely helpful for symptoms of Parkinson's disease. We have other classes of medications like dopamine agonists. So these are meant to represent dopamine receptors on a brain cell. And dopamine agonists, I like to call them synthetic dopamine. It's not exactly the same thing as dopamine, but it functions like dopamine. And so these are medicines like pramipexole, ropinirole, rotigotine. These are medicines that go to the brain and they work on dopamine receptors. They functionally act like dopamine. And these have been around for many years as well. Pramipexole and ropinirole have been around at least since the 90s. Another class of medications, really just a medication, Amantadine. It's been around for decades, and we think that it works by increasing the availability of dopamine in the brain, which I wish I could say more about, but it's just frankly not all that well understood. But I give these as examples of different classes of medications that we have currently available and kind of the different areas that they're working on to increase dopamine in the brain. And one more before we move on, which is deep brain stimulation. This doesn't fit well into the cartoon because, quite frankly, we don't know that much about how deep brain stimulation works, but without a doubt, it can help symptoms of Parkinson's disease. And this treatment has been around for many years. Surgical treatment options for Parkinson's continue to be refined. Additional treatment options that work through different neuromodulation pathways are also being investigated, things like focused ultrasound, transcranial magnetic stimulation. And so I just wanted to kind of briefly give a lay of the land about what we already have available before we talk about the future of treatments for Parkinson's. And the first point that I wanted to make is that, before we discuss individual drugs, just the big picture is that it's a really exciting time to talk about treatments for Parkinson's disease. I mean, if you went back 30, 40, 50 years and looked at what we had available, it feels like you could count just on one hand the number of treatments we had for individuals living with Parkinson's disease. And now not only do we have all these treatments that we've already touched on available, but the pipeline is full of new drugs that are coming out. Last week when I was preparing this talk, I went to clinicaltrials.gov, which is the US government's kind of repository and index of all available treatments for different diseases. And I searched for Parkinson's disease. And last week when I checked, there were 2,999 studies going on for Parkinson's disease. And so yesterday I had to update the slide because there's been another 13. So we're over 3,000 available studies looking at treatments and trying to better understand Parkinson's disease. Now, I just give that as an example to say what an incredibly exciting time it is to be talking about this topic. For the purposes of this talk, I'm going to split future treatments of Parkinson's into two main groups. So the first bucket that we're going to talk about are drugs that treat the motor symptoms of Parkinson's. And so all the drugs that we currently have available, so including the ones I just touched on, Levodopa, dopamine agonist, Amantadine, rasagiline, selegiline, these are all drugs that treat the motor symptoms of Parkinson's disease. The second group of drugs that we're going to talk about are drugs that might actually one day slow down the progression of Parkinson's, or drugs that could potentially even stop or halt the progression of Parkinson's disease. Currently, obviously, we don't have any drugs approved by the FDA for this purpose, but hopefully that will be changing soon. So these are the two different categories we're going to talk about. Talking about treatment of motor symptoms in Parkinson's, for anyone who's taken levodopa, you know that levodopa can often help in a significant way the motor symptoms of Parkinson's disease. So symptoms like slowness, stiffness, and often tremor can be improved by levodopa. One of the problems about levodopa, though, is that as Parkinson's progresses, patients can often have a less predictable response to the medicine. And this can feel like the medicine's not working, or it's becoming less effective. But we know that this is, in fact, due to just progression of the disease. And over time, the response to a dose of levodopa might feel shorter and shorter, but this could also be interspersed with periods of overactive movements that we call dyskinesias. And it's thought that these motor fluctuations in response to levodopa are really a problem of how levodopa is delivered. I tell my patients that typically dopamine is produced in the brain and it stays in the brain. And in Parkinson's disease, when we're replacing dopamine by using, say, levodopa or Sinemet, that's a bit of an unusual way for the brain to receive dopamine, because now instead of dopamine being produced in the brain and staying in the brain at very low levels, we're administering larger doses by mouth of this levodopa. It's having to travel, it's going through the intestine, through the bloodstream up to the brain. We're doing it 3 or 4 or 5 times a day, depending on how often a patient takes medicine. And this delivery mechanism of dopamine is really different than what the brain's used to. Many think that this is the reason that individuals who take levodopa have these motor fluctuations later in the disease, because we don't have a way to deliver dopamine in the same fashion in which the brain is accustomed to. And so this has really been a large area of research into future treatments for Parkinson's, namely, how do we deliver dopamine in a more physiologic manner? So lower doses delivered in a smoother fashion compared to larger doses delivered in pulses. I was trying to think of an analogy to better illustrate this point. So here's the best I got. Suppose for a moment that you have your groceries delivered via Shipt or Instacart or one of these new services out there that will bring you your groceries on a regular basis. And suppose that you subscribe to the service and you are used to receiving a typical set of groceries delivered once every 7 days. And those groceries that are delivered, they last you for about a week, and then you have another delivery arrive. So what if all of a sudden the grocery delivery service stopped bringing you 1 week's worth of groceries every week, and instead they started bringing you groceries once a month, but they brought you 4 weeks of groceries that one time a month? So technically, on a monthly basis, you're still getting all the groceries that you were due, but the amount and the timing of that delivery is really suboptimal for many reasons, because you're used to receiving a smaller amount more frequently. And in the same way, it's thought that this might be the reason that some folks have poor responses to levodopa as the disease progresses, because again, the brain's typically used to getting a very small amount of dopamine in a almost continuous fashion. And now we're giving the brain dopamine in this delivery system of 3 or 4 or 5 times a day in larger doses. And that's just not what the brain is used to. And that can cause problems. And so, drug companies have been trying to figure out how can we deliver the drug in a better, more physiologic fashion. And here's an example of that. This is a drug that's being studied. This is called an accordion pill of levodopa. So the accordion pill has these layers of levodopa arranged in a special way inside the pill. And the idea is that this special accordion pill would release the medicine from the pill into the stomach and the intestines in a very slow fashion, and that might help increase its absorption. So they had a phase 3 of this study back in 2019, and it did show that it helped symptoms of Parkinson's, but it wasn't necessarily superior to carbidopa, levodopa, or cinnamon. And the last that I saw, they were planning to repeat another phase 3 study of this drug to see if they can refine the process to better deliver levodopa. But this is just an example of testing that hypothesis that if we could improve the delivery of levodopa in a more smooth fashion, then maybe that might better treat symptoms of Parkinson's. Another way that they're trying to test this hypothesis, and most of us know folks that have type 1 diabetes and they require an insulin pump to treat their diabetes, and they have these little cartridges of insulin. They put the cartridge in the pump, and this pump delivers a very low, slow dose of insulin throughout the day. And most patients with diabetes even have one of these sensors now too, that's constantly measuring their blood glucose level and provides a feedback loop to the pump. So folks have asked the question, why couldn't we do this in Parkinson's disease with levodopa? What if there was a subcutaneous levodopa pump that similarly pumped levodopa into the skin or soft tissue and allowed this kind of slow, steady delivery of levodopa? That way, that would be kind of clever, right? And so they've been working on this for several years, and I believe are still in a phase 3 study of the system. But I bring it up just as another example of how researchers are trying to figure out ways to better improve the delivery of levodopa to better treat Parkinson's symptoms. Two more areas of research in treating motor symptoms before we talk about disease-modifying treatment. So one, better providing rescue treatments for off symptoms. We've talked about some of the complications of advanced Parkinson's, and we mentioned that as the disease progresses, the symptoms respond less predictably to levodopa, and some patients develop these motor fluctuations where they fluctuate between on and off time. And when on, motor symptoms are well treated, the slowness and stiffness are improved, the tremor is reduced, and when off, all those motor symptoms return. We've already seen some treatments be approved for these purposes in recent years to improve off time. Some of you may know about apomorphine that can either be injected or placed under the tongue to rapidly treat off times. We've also had a recent treatment approved in the last year or two, a treatment with inhaled levodopa that can help get levodopa absorbed and up to the brain more quickly and can rapidly help patients turn on from off times. And so these rescue treatments are still a focus. How can we rapidly help patients who are off turn back on? Another focus, another complication from long-term treatment of Parkinson's disease are dyskinesias, or extra involuntary movements. And so research has been ongoing about how to better treat those. There are at least 2 drugs that I know of that are being looked at to lessen dyskinesias, and both work by blocking neurotransmitters. So dipraglurant in phase 2 trials, that's been used to lessen dyskinesias by blocking glutamate, which is another neurotransmitter, and also a medicine called eltoprazine, which is also in phase 2 studies trying to lessen dyskinesias, this time by blocking serotonin receptors. So research continues to be ongoing. But even more exciting than talking about treatment of motor symptoms, I wanted to talk briefly about how we could potentially modify the course of Parkinson's disease, so slowing or potentially even stopping the progression of the disease. So this is a cartoon that shows a lot of things that are happening at a cellular level. And I want to talk a little bit about what happens in a normal, healthy cell before we talk about what can go wrong in Parkinson's disease. So this is a normal, healthy brain cell on the left. And there's lots of things going on. It's almost like a little city. So you've got lysosomes up here at the top that are responsible for removing trash and recycling waste in the cell. You have structures called the endoplasmic reticulum. So that's what's illustrated by this cartoon over here. And that's responsible for the transportation system within the cell. So moving things about the cell. And in a normal healthy cell, it's working well. We have mitochondria in the cell that are responsible for providing energy. And then we also have synapses so that the cell communicates with other cells. And all these things are working appropriately like they're supposed to, and the cell functions like it's supposed to. Now contrast that with this cartoon on the right, which is made to illustrate a neuron, a brain cell in an individual with Parkinson's disease. So there are several things that aren't working like they're supposed to. These lysosomes, the waste disposal system, if you will, it's not functioning like it's supposed to, and so waste is accumulating. The transportation system, the endoplasmic reticulum, is not working like it's supposed to, so things aren't being transported like they're supposed to. The energy cell, the energy part of the cell is malfunctioning. And the synapses down here, the cell can't communicate with other cells. Those are disrupted. And the take home from all of these processes happening is that you have accumulation of these little yellow tangles. You see these little yellow spindles in the cell that are accumulating inside and outside the cell. Those are meant to represent accumulation of a protein called alpha-synuclein. And in Parkinson's disease, alpha-synuclein is the protein that's associated with the disease. Rather than a cartoon, this is an actual photograph of a microscopic view of those alpha-synuclein proteins. And this is the protein that can lead to cell death in Parkinson's. And all of this background is to say that probably the most discussed way that's talked about to modify the course of the disease in Parkinson's is trying to figure out ways to reduce the accumulation of this protein, this alpha-synuclein. Again, because we know alpha-synuclein is the protein that accumulates that's not supposed to. And when it accumulates, these aggregates are called Lewy bodies. Some of you may have heard the term Lewy body before. And the hypothesis is that if we could somehow prevent accumulation of this protein, or if we could remove the accumulation of this protein, we could slow or even stop the progression of Parkinson's. And so this is being investigated in a couple of different ways. Again, this is the cartoon. We have this little yellow protein that's accumulating. It's misfolding and aggregating, and it leads to cell death. And so how do we prevent it from accumulating, or how can we remove it once it has already accumulated in the cell? And so one strategy has been to try and create antibodies against alpha-synuclein. So what if we could create an antibody that could tag this abnormal protein and basically tell the body's immune system to clear it from the brain? So, we could go to the lab and make this antibody and then put it in an infusion and give it to a patient with Parkinson's. And then these antibodies could go to the brain and help clear the protein. And so you'd basically be teaching the brain to create this immune response to the protein. And a very similar idea, but a slightly different pathway, is what if we just made a vaccine against this protein? So what if we took a little piece of alpha-synuclein and put it in an injection and give it to someone and basically teach the patient's body to create an immune response against that protein? And so then as the protein accumulates, the body is able to recognize that protein as being foreign, and it clears it from the brain. So you'd be essentially making anti-alpha-synuclein antibodies, which is a pretty neat idea. So that's one big focus of potentially trying to create antibodies against this protein that could prevent the progression of the disease. Another very important angle of disease-modifying treatment is what's the role of inflammation in the brain in individuals with Parkinson's? There's a lot in this cartoon, and we're not going to go through it, but I just wanted to make the point that there are a lot of markers of inflammation in the brains of individuals with Parkinson's disease. And I don't want to spend too much time going through this because it can quickly become disorienting, but suffice it to say that in the brain of an individual with Parkinson's, there are lots of markers of inflammation. And so this leads to some interesting questions like, does Parkinson's disease in and of itself cause inflammation? Or does Parkinson's cause some other process to happen? And then these inflammatory markers that we see, that's actually just the body's response to the initial process of the disease. So in other words, is inflammation the chicken or is it the egg? And research is still trying to answer this question. We definitely know there's inflammation in the brains of individuals with Parkinson's disease. So what if we could block that inflammation somehow that occurs? And this is an area of interest, just like folks take anti-inflammatory medications to treat other non-neurologic diseases. Maybe there's some kind of anti-inflammatory that could be given to treat neuroinflammation. There is one class of drugs that's already being studied to do that might reduce inflammation in Parkinson's disease. These are called Glucagonlike Peptide 1 agonists, or GLP-1 agonists. So these are actually medicines that are currently used to treat type 2 diabetes. You might recognize that glucagon. It sounds like glucose. So in diabetes, these medicines work by tricking the body, so to speak, into releasing insulin. But there's also been some studies to suggest that they might help reduce some of the inflammation that's seen in the brains of individuals with Parkinson's. And so this continues to be studied. We talked earlier about all of the processes at a cellular level that can misfire at a cellular level in individuals with Parkinson's disease. So we talked about transportation machinery or energy production or the cell's ability to communicate with other cells. One of those processes was the waste disposal system, and I mentioned that that's carried out by these small organelles called lysosomes. So again, lysosomes, they're small parts of cells, and they're responsible for breaking down the waste in the brain. And if any of the parts of those lysosomes don't work like they're supposed to, you can have waste that can accumulate in the brain. So these are two photomicrographs of two brain cells. So the one on the left is from a brain of a patient without Parkinson's disease, and then this image on the right is from the brain of someone with Parkinson's disease. And you can see all these yellow circles on the right, these little dots. And what these are these clogged lysosomes. They're clogged garbage cans, you might think of them. So the trash is not being disposed of like it's supposed to, and these lysosomes are kind of clogging up. Whereas on the left, you see a few of those, but in general, the trash system is working appropriately, as opposed to on the right where the trash, so to speak, the waste is accumulating, and that can have downstream effects. There is a particular protein that is necessary for lysosomes to do their work. Very rarely that protein can have a mutation in it, and when it does, it can lead to a variety of diseases. But it was found kind of incidentally along the way that when someone has two mutations in that protein, that there's a very small but a real increased risk of Parkinson's. And so some have thought, I wonder if that protein is somehow responsible for the progression of the disease in Parkinson's. And I wonder if we could somehow improve the functioning of that protein. Could we improve the function of lysosomes and kind of overall improve this waste disposal system and maybe prevent the progression of the disease? So there's a lot of research going on right now trying to enhance the functioning of that protein, or maybe even create a drug that might go to the brain and provide some other alternative garbage disposal system separate from the lysosomes, if you will. I know we're almost out of time, but I wanted to briefly just mention LRRK2. LRRK2 is a gene that when it is mutated, it can cause Parkinson's disease. And it's autosomal dominant, meaning that only one of the two genes needs to be mutated to develop Parkinson's disease. And this gene produces an enzyme by the same name, LRRK2, and that's what this cartoon is of the enzyme. And it's responsible for a lot of things in the body. It's responsible for building other proteins. And interestingly, when it's mutated, it becomes overactive, and it can cause Parkinson's disease. And that's not really well understood. It's not known whether that has something to do with that protein, alpha-synuclein, accumulating, or whether it's due to neuroinflammation. And so this is one more area of investigation is how can we reduce maybe overactivity of this enzyme and thus slow or stop the progression of Parkinson's. So my takeaways from this talk are, number one, there are a lot of potential treatments that are being studied. It's an incredibly exciting time. Lots and lots of studies going on. So a lot of hope and excitement about what the future will hold for treatments. We talked a little bit about treatments for symptoms of Parkinson's. So drugs that could improve delivery of levodopa, so providing levodopa in a smoother, more continuous fashion that the body's more accustomed to. We talked about treating off times that can help, drugs that might help patients turn on more rapidly, and also drugs that might lessen dyskinesias. And then here in the second half, we talked about treatments that might alter the disease course. So drugs that could potentially remove this abnormal protein from the brain, alpha-synuclein, drugs that might reduce inflammation, and also any drugs that might be able to improve kind of the waste removal system from cells in the brain. So thanks so much for tuning in. I've enjoyed talking about this today and learning more, and I'm happy to take your questions here in just a little bit.